you, Marissa. Can everyone hear me okay? Take that as a yes. All right. So, I'll just dive in right away. I'm here from the Department of Surgery, as Marissa said. I've been doing metabolomics for over a decade at this point. I started out doing NLR, studying hemorrhagic shock and severe traumatic injury, and have since transitioned over to using mass spectrometry. And I will tell you a little bit more about that. Just to go through real quick what you're going to hear from me today, I'm going to introduce sepsis to you, tell you a little bit about what it is, describe why clinical trials fail in sepsis, and let you know a little bit about what metabolomics can tell us about patient heterogeneity. First, before I start, I'd like to thank the Department of Surgery for their support in this research, and also, of course, GMSP for their help in collecting the data that I'm going to show you. I don't have any disclosures. So, sepsis is a response to infection that arises when the body's response injures its own tissues and organs. So basically, you get an infection. If you have a dysregulated host response, you can wind up severely ill with the systemic inflammation that will lead to organ dysfunction and can end with death. Many patients experience long-term physical, functional, and cognitive disabilities. And like most serious diseases, early recognition and early treatment are key to survival and better patient outcomes. And if you take a look at this figure here, you can see that this is a systemic, a truly systemic disease that affects all major organ systems, as well as metabolism, signaling. And so I think this is a disease that really benefits from a multi-omics approach. Briefly, sepsis is very impactful for the United States. Every year, more than 1.7 million people develop it. Nearly 270,000 American adults die every year as a result of sepsis. And it's quite expensive, with more than $62 billion spent on sepsis and healthcare costs. The Department of Health and Human Services has a specific Solving Sepsis campaign, and if you want to know more, you can look at their website. So sepsis is defined. We're on the third international consensus definition for sepsis as a variable and heterogeneous disease. It's been through many rounds of definitions and updates. We updated it in 2001 and 2016. And it's important to note that sepsis does not have a validated standard diagnostic test. It is instead a constellation of clinical signs and symptoms with significant biological and clinical heterogeneity. So sepsis is difficult to diagnose because of this variability. So there is no single clinical measure to diagnose sepsis. Instead, people look for systemic inflammation with high dysregulated temperature, heart rate, respiratory rate, or white blood cell counts. Some people will also look at markers for systemic inflammation. The SOFA criteria, which we'll look at in addition to oxygenation and coagulation, we'll look at liver function, heart function, neurological function, and renal function. And while these scores are validated and standardized, there is really not a the practices vary by institution, and how people act on the information from these scores also varies by institution. So I've mentioned that sepsis is difficult to diagnose. It is also difficult to treat. In fact, there is no standard treatment for sepsis. Instead, patients are monitored extensively and given supportive care in the form of fluids, antibiotics, organ support, and vasopressors. Now, many people have tried to cure sepsis. There have been over 100 clinical trials, and the result is that there are no treatments for sepsis to this day. So why do clinical trials fail in sepsis? I'll tell you a little bit about this study that was published in 2018. Many people were really excited about it. It was a relatively simple intervention for a complex disease, in thinking that sepsis is a product of a dysregulated immune system. The thought was that a simple immune support with high-dose vitamin C given intravenously could help patients. And instead, this clinical trial, there's another one in a long string of clinical trials that failed. And there were no differences in the outcomes that were assessed in this trial. And basically, like most clinical trials, the group looked at a standard population and randomized people to receive one of two interventions. So pretty straightforward and standard clinical trial design. But I have been setting this up as sepsis is a heterogeneous and complex disease, right? So if we think about it, what these clinical trials are telling us in their failure is that in my mind, they're demonstrating very clearly the complexity and heterogeneity of sepsis, and they need to target interventions to the right subset of patients. Sorry, this thing keeps just changing live on its own. So at the same time that this vitamin C study came out, this study on sepsis phenotypes was published. And, I've heard a lot of criticisms for this study since then, but I was very excited to see it because it was one of the first widely published studies to really demonstrate that there is no standard sepsis patient, that instead people fall into, could maybe be categorized into different phenotypes that have different presentations of the disease, require different treatments, and can present with different outcomes. And so if you look here, this group used clinical data to derive 4 phenotypes with different patterns in complications. They also went on to describe what different treatments these phenotypes might require. So instead of the approach here on your left, where we do a simple clinical trial, where we randomize people to receive 1 of 2 treatments, either a study drug or a placebo, and we see what happens. Instead, we're thinking more that a stratified approach is needed. I think most of you are probably familiar with the idea of precision medicine, where you're taking a much closer look at the individual patients. You're doing more precise screening of these patients to figure out what kind of disease, what kind of treatment is best suited to this particular patient. And with a disease as complicated as sepsis, this is really quantify 30 to 50 metabolites per sample. So if you really want to dig into pathway analysis and enrichment analysis, it's not great coverage of the metabolic pathways that may be of interest. So I wanted to move into using mass spectrometry, and I worked with the facility back in 2015, 2016 to do a little pilot study where I followed 10 individuals for a 24-hour period. At the time, I was interested in understanding more about circadian rhythms in critical illness. But the best science always happens when you get an unexpected result, right? And so I see this thing which interests me more than the circadian rhythm. And so what we're looking at here is a principal component analysis of this large untargeted metabolomics data set that we collected. And you can see each dot represents a sample from an individual, and the colors represent the individuals. And all of the healthy controls are clustered in that circle, kind of on the side of the graph. And they all look pretty similar to each other and very different from the ICU patients. What really surprised me was this patient-level resolution in the ICU patients, where you can see, these individuals, patient 7, patient 8, and they're all very different, but they're only like each other. So that really interested me, and I wanted to know more about that. And so after a while, I was able to do another pilot study where I collected samples every 8 hours instead of every 4 hours, and wanted to see if I could replicate this. And so by the time 2022 rolled around, CMSP is using biocrates kits pretty regularly, and I was excited to get this data, and I was really excited to see if it could replicate these patterns that I had seen in the uncharted data. And lo and behold, much to my surprise, honestly, I did see this patient-centric clustering again, and yet again, these patients are, that each dot represents a sample draw from a patient, and the colors represent the patient. And so, I'm very encouraged by this, but, I want to know more. And, I think, again, this almost raises more questions for me than it answers. But I want to take a step, since we're talking about biocrates and how great it is. I want to give you an example of this metabolite, isopC-18-2. This was a metabolite that I had given a putative assignment in my data set, my uncharted data set, because it wasn't, this particular feature was not in the data repository for CMSP, and so I was, giving this sort of a tentative assignment, but, seeing this clear difference between healthy and ICU patients, and also a significant circadian rhythm that was present in healthy controls, but not ICU patients. So in my next data set that I collected with biocrates in 2022, of course, I go look for this metabolite again right away. And, I'll never know if these two things are the same thing, I guess, but it is very nice to know what I'm looking at, so I can see a feature that's interesting, and I don't have to guess about what it is, and I can use it in downstream analyses. So the next phase of my research, I wanted to look more into longitudinal data, which I can then relate to patient outcomes. So this is data that I have just gotten fairly recently, and we are still in the process of collecting all the clinical data to go along with this. So I'm just showing you a real quick overview of this data. So I enrolled ICU patients and followed them at the sampling time point that you see on the side here. Basically, as long as folks were in the ICU, I was collecting a sample according to this timetable. And we do see that this pattern of sepsis patients are being distinct from healthy controls in the metabolomics data, and so that's very encouraging. I would expect to see that. And just doing a little quick dive into this while we are waiting for the rest of our clinical data. I'm just doing a quick view of the healthy patients versus the sepsis patients, and restricting it rather conservatively to only the metabolites that have no missing values. We can see that there are 127 out of 236 of these metabolites are significant after FDR adjustments. And the wonderful thing about biocrates is that since I know what I'm looking at, I can take this list of 127 metabolites and do a downstream analysis of them. You can do enrichment analyses, pathway analyses, cluster analyses, whenever your heart desires. So for this particular workflow, I have put that list of metabolites into Metabolomics, which is a website that many of you are probably familiar with. Did a quick enrichment analysis, and we find lots of interesting and relevant pathways. And it makes a fair amount of sense to look at these. I'm curious about other software recommendations and mapping to HMDB IDs. And so those are some things that we can discuss if you like. The future directions for this project, I will be getting transcriptomic data sets on these same blood samples and a robust clinical data set for all longitudinal data, so that we can better characterize the heterogeneity of sepsis and link to better identification of patient outcomes. And the biocrates platform, in my opinion, really facilitates this kind of integrative analysis for all of the reasons that Candace went over. And just to be very clear about what I think the advantages of biocrates are, is, as I said, I've been doing metabolomics in different platforms for a long time. With this kit, you get the sensitivity of mass spectrometry without the uncertainty of putative identifications that you're taught to do with an untargeted data set. You get water-soluble metabolites and lipid metabolites in the same data set without separate sample preparation. You can't do that with NMR. You get named metabolites, so you know what you're looking at, and that facilitates pathway and enrichment analyses. And I think that I would just stop there. I had a couple of other slides that I just don't think I'm going to go through right now, so we can do questions instead.